This is Amanda Welch welcoming you to this Bite-Sized Bio webinar, which today is sponsored by Qiagen. Qiagen is a Netherlands-based holding company, is the leading global provider of sample-to-insight solutions to transform biological materials into valuable molecular insights. Qiagen sample technologies isolate and process DNA, RNA, and proteins from blood, tissue, and other materials. Assay technologies make these biomolecules visible and ready for analysis. Bioinformatics software and knowledge bases interpret data to report relevant, actionable insights. Automation solutions tie these together in seamless and cost-effective molecular testing workflows. Qiagen provides these workflows to more than 500,000 customers around the world in molecular diagnostics, human healthcare, applied testing, forensics, veterinary testing and food safety, pharma, pharmaceutical and biotechnology companies, and academia, life sciences research. Today's presentation is titled A Start-to-Finish Guide to Target Gene Validation Using Quantitative RT-PCR and is being presented by Matthew Mule from the University of North Carolina School of Medicine and the NIH Oxford-Cambridge Scholars Program. Matthew is a graduate student at the University of Cambridge and the NIH Oxford-Cambridge Scholars Program. His current focus is discovering core molecular signatures of the immune response by integrating next-generation sequencing, single-cell transcriptomics, and flow mass cytometry data. Previously, Matthew worked at Tufts University as an undergrad, at the NIH as an RITA fellow, and UNC Chapel Hill Medical School. Now, as always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen. I'll put them to Matthew at the end. The recording of the webinar will be available at bit.ly/targetgenevalidation. That's target gene validation, all one word, lowercase. So now over to you, Matthew, for the presentation.
All right, thanks very much. I'm just going to jump right into it. So I just want to do a quick disclaimer that I'm not affiliated with Qiagen or any company or anything like that. I'm a graduate student. I've been doing gene validation with qRT-PCR for a long time. So the reason I'm doing this is to just share some knowledge and also distribute some resources. So everything I'm going to show you at the end is on a GitHub. So all my analysis, all of the Excel spreadsheets, the R documents and everything like that. So anyone can just download them and go through the webinar tutorial basically again. So if you don't retain everything this first time, that's okay. So basically the goal here is to allow you to understand how to do this. So if you have a list of genes that you want to validate their expression levels or their mRNA quantity in cells, and you don't know how to do this, the goal of this tutorial is to just tell you from start to finish, like how would you go through, design an assay, set up the experiment, analyze the results. So, and part of that is going to be some walkthroughs where I'm going to show you exactly how I would go about doing this. So typically, but what I'm sort of basing this off is, you might have some high throughput data, you might have microarray RNA-seq data, and you have this list of differentially expressed genes, and then maybe you've done some pathway enrichment or other gene set enrichment analysis, and now you have a list of genes or a pathway that you want to validate. This is a side note. Again, as a resource, I link to a lot of publications in this. So, and because this tutorial will be available online, you can go through and look at all these. But this is just this is outside the scope of this tutorial, but it's an interesting paper on sort of the correlation structure of genes that you should be aware of if you're trying to validate large gene sets. So the number of genes that you want to validate is going to sort of dictate the experiment design, right? So if you have a handful of genes, it's probably feasible to go ahead and first look online to see if there's an assay available anywhere, but it's probably feasible to design the assay yourself, optimize it, and run the experiment. When you get in the level of 20 or any more than that genes, you're going to need a more sort of what I would call a medium throughput approach, where there's these plate-based assays or microfluidics assays that allow you to measure many genes simultaneously with qRT-PCR. And these are usually a bit easier overall to use because you don't design the assay. So now I'm just going to give a brief overview of qRT-PCR. So it's called a bunch of different things, but basically at a high level, what I'm sort of basing this analysis off and this tutorial, but obviously the sort of overall schema scales to anything. So you have some control cells, maybe you treated a mouse with a drug, you isolated some tissue of interest and extracted mRNA, you're gonna make cDNA and add it to a plate with some gene-specific primers and quantify the sort of inverse relationship between the cycle number at which the fluorescence hits some threshold level and the starting amount of cDNA that's specific for your gene target. So I'm going to go into that in some more detail now. So I think it's really helpful to sort of marry these two. Sort of, you have a conceptual model of what PCR is, which is that you have some target and you have Taq polymerase that is amplifying some primed specific sequence that you're interested in, and every cycle your PCR target gene doubles in concentration. So that's good for conceptually, but in order to sort of understand quantitative PCR, it helps to sort of think about it in this sort of more formal framework where we can think about the number of molecules of DNA we have at the start of the reaction, right? So this is basically the equivalent of the expression level, we'll say, of that mRNA in that cell population you isolated the RNA from. That is equivalent or is proportional to the number you get at some cycle will be proportional to that number multiplied by 2 to the power of whatever cycle number of PCR you're at. So, for example, if we are at the third cycle, right, the number of molecules we have at the third cycle here is proportional to the number we had in the beginning multiplied by 2 to the power of whatever cycle we're at. So this is also making the assumption that every single time we amplify and we go through and we do our PCR reaction, we double the number of molecules. And so that is actually an assumption that you need to validate experimentally, which is tied to the efficiency of the reaction, which I'll go into more in detail in a bit. So when I say, the cycle at which we can detect some level of fluorescence, what I'm talking about is this thing, the cycle threshold, right? So for every PCR cycle, you're accumulating target, and at some point, the accumulation of target, which accumulates fluorescence, will reach some level that is above what you said as this threshold level, this baseline level of noise, and then you get amplification above that level at some threshold that you set, and that is the quantification level for that that specific PCR reaction. And so I'll refer you to this publication that was actually on Bite Size Bio for a little bit more about CT values, but that is what we are actually quantifying. So I'm going to talk now about designing an assay. So this is, it's hard to do this without sort of showing you, so I'm just going to kind of show you, but just a brief note on should you even design an assay, right? Okay, so if you're going after this WT1 gene, and there's this publication where this consortium has comprehensively gone through and measured every single known assay for this gene and validated the best one for a clinical test, you'd be crazy to try to make another assay for this. Just use the one they validated, right? However, if you're after some sort of novel splice variant in a gene, that's not necessarily going to exist in some off-the-shelf assay. So you're probably going to have to design that. So this just, again, is sort of a resource, but what I'm going to do now is sort of walk through an example. So this is telling you exactly what you need to do. I'm just going to show you. So for designing primers for your assay, there's really no shortage of primer design, but what I'm going to do is use Primer Blast, which is a NCBI tool, and it sort of integrates Primer 3, which is a very popular primer design algorithm, with blasting that sequence that you pull against the genome to make sure that the primers are specific. So what I'm going to do now is just show you how I would do this. Okay, so let's say we are interested in this gene. Okay, so this is the TAP1 gene. What can we do to sort of learn about it and to see how we're going to design this assay? Okay, so I'm interested in TAP1. I am going to go to the NCBI gene database, or you can go to the nucleotide database to pull a cDNA sequence. But basically what you're going to want to do is go down and, okay, let's say you're interested in whether or not this gene is expressed in a certain tissue. Well, you can sort of learn a little bit about that just by NCBI's website, where they have this bio project, which you can look into. And this is RNA-seq reads for a given number of samples for this gene in all sorts of different human tissues, which is pretty interesting. So already right there's some interesting data you can learn about your gene. But anyway, what I want to draw your attention to is, okay, so this is our gene sequence, and these darker regions here are going to be the exons, and these light regions in between are the introns. Okay, so if we're designing a qPCR assay, there's pros and cons to sort of every sort of design, and this is a sort of long conversation, but to sort of give you the gist of it, if we have a assay that amplifies just one single exon, the one issue that can arise unless you extensively validate it is that you could also be amplifying genomic DNA. However, if we expect our PCR product to be, let's say, 150 base pairs, and we have one gene-specific primer here and another one here, then we're going to know that if we get that size target in our PCR reaction, then we know that we're not amplifying this genomic region. This is huge, thousands and thousands of nucleotides here. Another thing you can do is I want to draw your attention to these blue sort of histograms here you see. These are actually RNA-seq coverage reads. So they correspond with the consensus cDNA sequence here, which you can see. But also something to keep in mind, and something that's kind of interesting, is that there are these reads that are in these intron sequences, which may be noise, but they also may be splice variants, which could be interesting and something you might want to look at. So, okay, so let's pretend we want to design an assay for this sequence. I can click on it and then click primer blast. Boom, super easy. Okay, so then we're going to get this primer design tool where we can go in, and then we are on the RefSeq mRNA database. There are all sorts of parameters that'll allow you to take a look at. But again, one of the main things that I was talking about was whether or not you want to be on an exon-intron junction and the number of mismatches, et cetera. So then all you do is just get primers. And what this is going to do is find optimal primers at a given TM that you can specify. A given melt temperature of the assay that you can specify. And sometimes this takes a while, but I've already run it for you. So these are the results. So it shows you the location of the gene-specific primers, and then it gives you the sequences here. So yeah, so this is a great tool to design an assay. Another thing you can do is add a probe. Okay, so probe-based assays, and there's similar design parameters that you want to consider, and you'll be within this software you can specify that you want to design a probe as well. There's other software, for example, this AutoPrime. This is another method you can use, and you can just actually enter in a gene symbol, which is pretty cool. And one thing to keep in mind is they're not going to be necessarily the same. So you might get different results with AutoPrime and different results with doing this primer 3. But really, you just have to pick something. The most important thing is to make sure that it's specific for your sequence, and you blast it against the genome, which I like the fact that this does this automatically. Okay, so that's sort of a brief overview on how I would sort of go about designing primers. So another thing to keep in mind, again, this is sort of outside the scope of this presentation, but if you're validating RNA-seq data, you might want to run your reads through a program called MapSplice. There's a couple of programs that do this, but this is sort of the best supported one, which looks for specific novel splice variants in your data, which could be really interesting. So evaluating the assay, let's say you've ordered that assay and you have it, came in the mail. All right, now what are you going to do? So I'm going to come back to this conceptual model and mathematical model of PCR. We need to, what I said before about, okay, the number of molecules at a given cycle is proportional to the number in the beginning times 2 to the cycle number, but that is assuming that you have 100% efficiency. So you need to validate the fact that every cycle of PCR, your target doubles. And so the way you're going to do that is basically you can run a sort of standard curve experiment, or let's just say, well, to back up a little bit, let's just say we run 20 nanograms of cDNA, 40 nanograms of cDNA for our target sequence, and 80 nanograms of cDNA, right? And then we add our assay and we do our qPCR reaction and we get these cycle threshold CT values, right? Well, what we would expect then is that for each one of these starting concentrations where there was double the amount of starting product, we would expect the cycle at which the fluorescence accumulated beyond that threshold level to go up by 1, okay? So that's sort of, that's the sort of conceptual model, and you can prove that to yourself with this formal model that the efficiency of the reaction is intrinsically tied to this idea of, every time you double, or for every cycle, you are doubling the target. Okay, so, and you can calculate your efficiency by simply, regressing these points and building a linear model, and the slope of your line is equivalent to that efficiency. So, another way you can do this, and sort of you kind of kill two birds with one stone actually when you do this, is you can create a serial dilution experiment where you do 10-fold concentration differences over a wide range of concentrations. And what this will allow you to do is to build that same sort of linear model, but you also figure out the limit of detection of your assay. So if you have, all the way down to what you think is maybe 10 copies of your starting sequence in the starting reaction, you can sort of know whether or not your assay is able to detect that low starting amount. And so the absolute limit of detection is going to be equivalent to the y-intercept, okay, if we regress the concentration against the CT values. And you can sort of prove that to yourself. So another thing you're going to want to do is you're going to want to make sure that the temperature that you run the extension step in the PCR matches whatever you predict by some algorithm that it says it's going to be, right? So when we designed those primers, we said, I want the extension step to be 60 degrees. So you could run out different annealing temperatures, okay? And you can sort of get a sense of where you have sort of the maximum fluorescence detection. And you can do this with a standard PCR and just do a gradient. And so this can allow you to narrow in, maybe you designed it to be 60 degrees, but actually at 64 degrees, it's a much stronger band and you get a single product, okay? So on the note of single products, another thing you're gonna definitely wanna do is, before you go into the qPCR, you're probably gonna wanna just take your cDNA you're trying to amplify, add your target assay, run it out on a gel, okay? And so if you see these multiple bands, you know right away that your assay, something's not right, right? You're amplifying multiple products. Similarly, you can do a melt curve, okay? So this is something that it's an option on the qRT-PCR thermocycler, right? You just say, it's gonna say, do you wanna run a melt curve? Say yes, okay? And so when you're doing those efficiency calculations, what you also want to do is run a melt curve. And what this is going to do is at the end of the reaction, it's going to slowly increase the temperature of the actual reaction. And what this melt curve plot is sort of it's the negative time derivative of the reaction as it's melting. And so you get these peaks, which correspond to the dissipation of fluorescence for a single product if you have a single peak. However, if you have two peaks in this melt curve, that you're going to need to sort of troubleshoot that. And it's possible that you still only have one peak, although it's rare. You still have one product when you get these multiple peaks. It's rare, it's possible, but I'll sort of refer you to this article here if you want to learn more about why that might happen. Okay, so now just to talk briefly about the importance of a control gene. So a control gene, or a housekeeping gene, you might hear it called, this is for normalization. Okay, so we're normalizing the expression levels to different amounts of total RNA produced by the cell, different cell numbers, different cell populations. Okay, so if you were to just look at this data here, where I have on the x-axis, I have our control group, and the cells that were treated with drug. On the y-axis, I have the CT. If you were to just look at the CT levels for our, let's call interferon gamma, this IFNG, as our gene of interest. Okay, if you were to just look at the cycle at which the fluorescence crossed that threshold for the control and the drug treated, you would maybe come to the conclusion that, oh wow, the drug treated samples, they reach that cycle threshold at a much earlier stage in PCR, so there's much more target, right? So this is, we would call this, overexpression, right, of this target gene relative to the control group. And so if you were to, just calculate it using that exponential relationship, and I'm going to go over this calculation later, you would maybe falsely, incorrectly conclude that this gene is being overexpressed 35-fold. But actually, if you normalize this to the level of the control gene, we see that also our control gene also was reduced as an effect of our drug treatment. So after we adjust for this, the fold change is 0. Okay, so how can this happen? I encourage you to sort of think about that. But one thing to think about is, it's possible that our drug treatment altered the cellular composition of our sample. So there's different cells that are there. So it's not really overexpression or underexpression that we're detecting. We're just detecting different cell populations as a result of our treatment. That's like probably the most common thing you have to think about when anytime you're doing bulk transcriptomics or bulk targeted gene expression assays. And then, also think about what would happen if our housekeeping gene was increased. It's like a good exercise to think about. So how do you select a control gene? Well, okay, so here I'm sort of plotting the sort of scientific integrity of 3 different things you could do as a function of sort of the effort and time needed to do them. So, beta-actin is like a commonly used housekeeping or control gene. So let's just use that. That's not going to stand up to review, and I would say that's a really not a good idea. Beta-actin can be expressed. It is not a good housekeeping gene, let's just say that, necessarily. So you need to find the housekeeping gene that's going to allow you to best assess changes in your genes of interest. And you're going to need a gene then to use as a control gene that doesn't change really and has low variance as a function of your control, your treatment scenario. Okay, so another thing you do is review the literature, and use what others have used in your experimental model. That's fine. That's fine to do. So if someone else has done this exact same thing you're doing and they've shown this system works and their data makes a lot of sense, then yeah, you can do that. Probably the best thing to do is test out a bunch of different potential control genes and pick the one that's the most temporally and biologically stable between these time points in the treatment and control groups, and validate some experiments using multiple control genes. So that is hard and takes time, but it's actually made a lot easier by what you can do is just order one of these housekeeping gene plates that have primers already lyophilized to the plate, and just add your sample and master mix, and you can get an idea as to which one of these genes has is the most stable in your experimental setup. And there's a way to there's a number of ways to assess this. Sometimes it might be very obvious. You might have huge error bars on if you were to just plot this gene as a function of all your different experiments. But there's actually a little bit more of a formal way to do this, and to do this, meaning to how to select this gene after you run an experiment like this. And I'll refer you to this really good publication here, which shows how to do that using this delta CT method. Okay, so doing the experiment. Okay, so let's say now you've got the assay, you validated it, everything works. You know, now what are you going to do? Okay, so you have to extract mRNA. This is, you know, it's like baking a cake. You just got to follow the directions. You just got to do it. I strongly recommend, you know, working in a room that is sort of, you know, if possible, or a space that you designate at least for that day or that week as your RNA workflow room, and you wipe everything down with one of these products like RNA Zap or some sort of RNA eliminator, and you're working quickly and everything is on ice at all times. Once you isolate your RNA, you're going to have to do some quality control on it. So there's a couple of different things you have to do. One is measure the optical density. So this is essential, actually. You got to, because you have to know the concentration of your RNA in order to make your cDNA. But another reason we do this is because, so, you know, nucleic acids absorb UV light, and so the amount of absorbance is proportional to their concentration, but it's also a way to tell if you have any residual phenols from your RNA extraction that are contaminating your RNA. And that's important to know because it can sort of affect the quality of your results. And there could be PCR inhibitors, for example, in that RNA that you're using. So, and the absorbance at 260/280 ratio for RNA is accepted as, you know, anything greater than 2. A little bit more of a quantitative way to see if your RNA is stable and in good shape is to run an RNA gel. And so this is a bioanalyzer run where basically we have the, it gives you this RNA integrity number, which is just an algorithm that's calculating, effectively, it's the ratio of these peaks of your 28S and 18S RNA. If you're in a human system, you can get a mouse system or other experimental systems as well. But this is a really good way to make sure before you spend a lot of time doing a bunch of experiments, if your RNA integrity number is like 2, then you're going to want to think about maybe re-isolating RNA if possible. So again, here's similar information, but I just plotted a couple different RNA integrity numbers. Like if it's 6 or 7, you know, it's probably fine, but it sort of depends heavily on your application. So now you're gonna have to synthesize cDNA. Again, this is just, you know, it's like cooking. You just have to follow a recipe, basically. A couple things to note is, I think it's interesting to think about mRNA is really, really rare in a given RNA sample unless you poly-A select it. So you're only getting about, due to the capture efficiency of like a cDNA reverse transcription reaction, you're only getting about 10% of the mRNA that's there. And that's only about 3% of the total RNA that's in a sample. So most of it's ribosomal RNA. So another thing to think about is if you're using one of these plate-based assays, one of these more, I guess, medium throughput approaches, you're going to want to use whatever cDNA synthesis kit they recommend, because sometimes, for example, Qiagen does this, there's these controls that are engineered into the cDNA synthesis. Another thing to think about is having a no RT or a minus RT control in your plate setup, which we'll talk about in a second. Also, spike in controls. Again, this is sort of outside the scope of this talk, but I want to refer you to this link to think about if that's something that you should do. Okay, so now you have everything. How do you actually set this up? What you're going to do, it's going to be probably a 96 well plate, and you're going to add, the first thing you're going to do is add PCR master mix. So if you're using a probe-based approach, or you're using intercalating dyes like Cybergreen, it's going to have some pre-formulated master mix. It's usually at about 2x. And so let's say you want a 25 microliter reaction volume, you're going to add 12.5 microliters to every well. Next thing you're going to add is your gene-specific assays that you have validated. So in this case, I'm just showing, okay, 2 genes of interest we're quantifying and a control gene. And so how much you add really depends. If you're buying one off the shelf, start with whatever they recommend and kind of go from there. But if you ordered specific primers, the assay final concentration should be about 1 micromolar, plus or minus a probe if you have that. So in this case, I'm just showing, gene of interest and a control gene. Okay, so after you've added that, you're going to add to your plate your cDNA. And so in this case, I've divided the plate in half. I have a whole half of this plate is devoted to a no reverse transcriptase control. And this is basically to see if we're amplifying any genomic DNA, if there's genomic DNA contaminating. So that's an important control to have. And again, I'll refer you to that article actually that was written for Bite Size Bio that actually goes into a good amount of detail and explains why it's an important control to have. Whether or not you need to do this in every experiment is up to you. If after several experiments and several RNA isolations, you see that every single time you run this, you get no CT values, then you're probably fine. But this is probably the best practice for setting up the plate. And now you're going to add your cDNA for each sample. And so in this case, I have sample 1, sample 2, sample 3, sample 4. Okay. So how much cDNA do I add? And another question people have asked me, who I've helped out with this, is, should I dilute my cDNA? And so it really just depends. So if it depends on how much you want to add and how easy you want it to be to add it. So basically, if you used 1 microgram of RNA to do your cDNA synthesis, you just have to, you can go through and read this, but if you add 80 microliters of water to that 1 microgram per 20 microliters stock, then you would add 5 microliters to each reaction to get 50 nanograms. So now you're going to run the thermocycler. You're going to choose a threshold. Often, you can just use the automatic threshold that the machine sets. One thing I'll caution you on is occasionally you will have some background noise, like this image is showing. And one thing to keep in mind is that you can have sort of background noise from adjacent wells that aren't even amplified. So for example, you didn't even have anything in there, okay? And there will be these random sort of spikes. Sometimes I've seen this, and it's not an actual amplification, okay? It's just random noise. And so you're gonna wanna eliminate those wells before you calculate the threshold, because if you have some weird noise over here, and it's like this weird wacky curve, that's going to affect how this threshold is set. I've never seen a case in which this matters, but some people say that you should use the same threshold for multiple runs. Probably certainly the case in sort of a clinical setting, but it's not too important. Okay, so now I'm gonna briefly go through sort of an example data analysis. So let's say you ran this experiment, you get the data, what do you do? So in this experiment, it's the same thing as I've set up on this plate that you can see here, okay? And so I'll refer, you can refer to this and go through this again if it's, I go too fast, but again, everything is sort of online. Okay, so I have I'm showing the analysis here, the way you would do it in Excel and the way you would do it in R. And you can go through these, and this R is in the R analysis, there's a written tutorial on exactly what you're doing at every step. So, but sort of very briefly, you're going to get this output from the instrument, and it's going to have these CT values, okay? You're going to average those CT values for each sample in each gene. So here I have the control gene. For sample 1, I have 3 replicates, and I'm averaging the values for each condition, the no drug condition and the treatment condition, okay? So this is how you would do this in Excel. It requires some subsetting and sorting the rows and everything like that. I also show how you would do this in R. It's a little bit easier. So for example, in R, you just have to type this. It's a couple of lines, and it automatically calculates this for you. So then you're going to calculate your delta CT values, okay? So this goes back to those earlier slides I was showing about so we are normalizing against the expression of our housekeeping gene. So in this example, we have our delta CT for our no drug samples, okay? So for this gene, for interferon gamma, for sample 1 in our no drug control, we calculate the delta CT as follows, right? So we have the CT value for our gene of interest, and we take that and we subtract the expression or the CT value of the control gene for that sample, okay? And we're just gonna do that for every single sample. And in this analysis, what I've done is, what I'm showing is the delta, the so-called delta CT, okay? So this is what this is just the log ratio, so the fold change in our treatment samples as a function of, or as a ratio of the expected amount we would see in the control samples. Okay, so what I've done to do that is average the no drug control samples for our 2 genes. In reality, you would want to do this on many, many more samples. And the analysis is going to be different if you have for example, match case control. Okay, so let's say you have the mouse at one time point, you treat it with a drug, you isolate tissue again at another time point. That would be a match case control in which you wouldn't do this averaging. But just for the sake of this analysis, I'm averaging the delta CT values for each gene of interest in the no drug group. And then I am taking the log ratio, so it's 2 to the minus delta CT. So basically what you have is, for the interferon gamma gene for sample 3 in the plus drug group, we have the delta CT value. We take that, we take 2 to the negative power of that minus the expected expression we would see in the non-treated group, okay? And that's going to give us a fold change value. And so it's 2 to the minus, this is just, log ratio, okay? So when you have the log, you're subtracting. And you do the same for sample 3 and sample 4. And now we can also do this, we can do the same exact calculation for the other gene of interest in sample 3, which is the plus drug treated, and sample 4, which is the plus drug. And now we have fold change in the drug treated samples of 2 genes normalized by the expression of our control gene and relative to the non-treated samples. So that is what this calculation is. Now, obviously, your calculation is going to depend on what it is your experiment is doing, but again, that's how you would do this in Excel. And you can see there's a lot of sort of referring to different rows, and I think it's a little bit, it's okay to do this in Excel if you have maybe 2 genes, but when you start to get a little bit higher throughput, if you're doing 10 or 20 genes, this becomes really easy to make a mistake. So I really recommend using R actually, and I've shown you how to do this exact same stuff in R here, and you can sort of use this as a reference if you're comfortable with it. And then, you would plot the results. So here we have the effect of the treatment relative to the average expression seen in non-treated samples normalized by a control gene of these 2 genes. And so I'll let you sort of go through that analysis again. And again, everything is going to be online. So yeah, so having gone through the analysis, again, I'll refer you to these slides if you want to take a look at what we've computed. And again, here we have group 1 versus group 2, and we're comparing group 2 to the sort of global average we would see in healthy controls, okay? Another way you could do this would be to have matched case control or matched pairs experiment. And so that's going to change the analysis slightly in that you wouldn't do that averaging step. You would just do the delta-delta CT of time point 1 versus time point 2, for example. So very briefly, sort of medium throughput approaches. These are going to be, again, you have a plate-based assay. All you have to do is add master mix and your sample cDNA, and you're going to really just run the plate as is. These assays are usually pre-optimized, they're ready to go, and you don't have to do any of that assay validation. And so it's really good if you have, many samples and, a lot of genes that you're trying to analyze. I would really recommend one of these more medium throughput approaches. And I would also recommend, if you're going to do that, it's going to be a lot more scalable to analyze everything in R if you're comfortable doing so. So again, finally, just a couple of notes on MIQE here. So this is the minimum information that you should publish for a qPCR experiment. And this is just a really good resource. Basically, these are the different parameters that you should show in a publication somewhere, whether it's in the methods or in the supplement. If you do an experiment and they are basically sort of grouped by whether something is essential or whether it's recommended that you report this. So this is a good thing to have. And then sort of finally, just some visualization examples. So this is from one of my publications, and whether or not you're interested in bone marrow transplant, it's a good resource just because it basically heavily relied on qPCR for this paper, and I have a lot of different visualization and analysis methods that are in the supplement, so I would refer you to that. And also, for example, here's one of those matched pairs experiments. So I have, each person, there's the treated and non-treated samples, and I've calculated the delta-delta CT for each matched pair. I have, another way I've shown the data is to reverse the delta CT axis. So I have negative to positive numbers. So just to see the, it's a sort of clean way to visualize fold changes. And I also have interpolated copy number using plasmid standards. So it's just another way you can do this that I didn't really go into, but I will refer you to that. So with that, I think I'm just about out of time. So I'll take any questions.
Thank you, Matt. That was an excellent presentation. Now, we have a few questions in the audience, but if anyone else has a question, please feel free to post it in the questions box that appears in the right name. So our first question comes from Varian, and they're asking about, does the efficiency depend on the enzyme you are using, or do you need to measure it for each primer?
Oh, I see, yes. So, no, the efficiency is really a function, well, sort of theoretically, yes, it's a function of the specific enzyme that is the Taq polymerase that's in the reaction, but what you're analyzing when you're analyzing those efficiencies is the efficiency of your assay to be able to amplify that sequence and double it with every cycle of PCR. So that efficiency calculation you would need to do if you have, let's say, you designed an assay for 3 genes, you would definitely want to do that efficiency calculation for each gene, because the efficiency for one PCR reaction using the same enzyme is going to be completely different from another one using that enzyme when the assay itself, meaning those primers, are different.
That makes sense. And they have a follow-up question. What do you do if the efficiency is not 100%?
Yeah, so that's a good question. It's there's a couple of things you could do, and if it's wildly off, so if you have like 50% efficiency, then you just shouldn't use the assay. So that's just, you're going to have to kind of go back to the drawing board. Let's say, you know, if the efficiency is less than 100%, one thing you can do, and I've seen this done a couple times, is when you go, or I can show that calculation, but when you are actually calculating these delta CT values, or these delta CT values, they rely on this, right? The fold change value relies on 2 to the negative delta CT. Okay, that's the calculation. If your efficiency is not 100%, and you can actually go and calculate what the efficiency is using these equations, and basically, you could adjust your fold change values to reflect the efficiency of your assay. So in other words, if you have a less than 100% efficiency assay, you can adjust this. So you would do, instead of 2 to the delta CT, you could do, let's say, 1.8 to the delta CT, if that makes sense, based on, and that again is based on whatever the efficiency you calculate is. So it might be 1.75, it might be 2.05. That's actually the most robust way to do it. But commonly, as long as it's within sort of 10% plus or minus, I usually don't see people actually change this power when they do the calculation. But I have seen it done before when someone had basically like an 80% efficient assay. They adjusted their fold change values to reflect that.
That does make sense, and because I'd imagine that small amount wouldn't factor in too terribly much.
Right, absolutely.
So we have a question about cDNA. Is there a way to check, to quality check cDNA?
Really, there's that's not really commonly done, actually. So you have sort of residual enzyme left over after that cDNA synthesis reaction, and there are some, and so basically for qPCR, people don't quantify the cDNA. There are ways to quantify cDNA, certainly, and you can get creative and use those ways in your qPCR reaction, and however, they're usually for like NGS technologies. When you have a sort of a cDNA library, you can quantify the quality of the library using different metrics, but usually that's not done for qPCR.
Okay. And then we have a question about the best practice to remove genomic DNA from an RNA sample. So some kits recommend that you use on-column DNA digestion at room temperature for 15 minutes. Others have other ways of doing it. Is the on-column DNA digestion, is that sufficient to remove most of the genomic DNA? Or do you have some other insight for that?
So what I would recommend is in the so if the cDNA kit includes a method to remove genomic DNA. So it's usually an enzyme treatment, right? I would do only that. In the past, when I've tried to add on, in addition to the genomic DNA cleanup assay that is part of the cDNA kit, if I've tried to add on the on-column DNase treatment or genomic DNA contamination removal, I've seen sort of gross differences. And this is just, personally, so you can certainly obviously test this out. But I've seen the CT values are affected, by 1 or 2 if I'm doing multiple genomic DNA removal steps. That's my experience. But I would basically encourage you to test it out. What you would do is just set up one reaction with the genomic DNA removal plus the on-column removal and just see if your results are affected by that. But yeah, if there's no genomic DNA removal enzyme treatment in the cDNA synthesis kit, yeah, I would recommend using one of those on-column digestion methods. Those work quite well. So yeah.
Okay, and then we've got, I think it's he asks, is there any difference in using linearized plasmid or oligos as standards?
So I've used both, and they both work. So basically, one thing, yeah, I would definitely, first of all, I would definitely recommend linearizing plasmid, as the question asker has said. So if you have circular DNA, the PCR efficiency is really low. But another thing you can use are these IDTG blocks, which they might be referring to as the oligos, but they're it's just a synthetic sequence that is made by like IDT, for example, or one of these companies, and they, generally they work really, really well. I think there's, in my opinion, sort of the differences doesn't really have to do with the PCR reaction efficiency or anything like that, or how you're able to quantify the reaction. It really has more to do with like the cost benefit of making and cloning a plasmid yourself, if you want to do that, or if you're ordering a plasmid versus ordering one of these so-called G blocks, or I think these long primers. Basically, it sort of just depends on cost, really, because I think they both work quite well.
Okay. And then we've got a question from Zane. Well, actually 2 questions. The first question is, what is the difference between CT and CQ?
Same thing.
Okay. That would be kind of fast. And then the second is, how do you determine threshold? I know that you talked a bit about trying to remove that background noise, but is there, could you talk a little bit more about that?
Yeah, you basically, so I can go up to, where's that picture I had? What you'll see, I would encourage people who are interested in this to just play around with the threshold that they use and see how it affects the results. So if you were to set a threshold down here, or you were to set a threshold sort of up here, how does that affect your results? And because it has to do with the distances between these points, once you're at this you're at this crossing point, it's not the threshold has a very minor effect on your overall results, as long as you're in this sort of linear part of the amplification curve here. So if you see that I'm on this straight point of this amplification curve, if you start to set the threshold way up here, then this fluorescence value is on its sort of peak phase, and so they might not be even across samples. Like this one might be still in its linear phase, and this one might be trending to the right a little bit. Okay, so but overall, it's really usually the auto threshold on most machines sets the threshold well, and then you can kind of just use the same threshold in all of your replicate experiments, and that's usually a quite robust way to do things. But yeah, again, you just want to make sure you're in the first part of this sort of linear phase of this amplification curve here, where it's not on the plateau and you're above the background noise. And that's what the machine is doing automatically. It's finding the optimal solution for that. But, you know, again, the only thing you have to keep in mind is if there's some noise in this plate, like that, you know, you got a little bit of master mix or something in one of the wells that you didn't even put anything in there, and it's this weird spike, that is going to mess up the threshold. So you want to just only select the wells that actually have any reaction in them when you're setting the threshold. So that's how you would go about doing that. But again, I would encourage you to sort of play around with, you know, export one version where you set the threshold up here, let's say, and and export one version where you set it down here. And I think you'll find that the results are quite similar.
Okay. Yeah, I've played around a bit with setting threshold as well, and found that the auto one tends to work best for me.
Yeah. Yep, same here. So.
And then we have, this is an interesting question. So Clement asks about, and maybe you have some insight to this. They say sometimes they get PCR efficiency above 100%. Do you have any idea why that might happen?
Yeah, that definitely happens all the time, and it's sort of I have to think about that a little more. Why you get so in terms of the actual theory, why you're getting greater than 100% efficiency. I'd have to think about that a little more. But yeah, that's a good thing to Google.
Yeah, it is a good question. And so then we have a question from Elsa. They're asking about what is better, the 2100 bioanalyzer or conventional PCR than electrophoresis or qPCR? Whew, that was a mouthful. Or they rephrase a little bit later. What is the reason for using the bioanalyzer than conventional PCR, than electrophoresis and also qPCR?
Are they, so is this referring to quantifying the, like the is that what I heard? The bioanalyzer? Is that
bioanalyzer, the 2100 bioanalyzer.
So the 2100 bioanalyzer is more for quantification of, or it's not for quantification, but it's more for QCing the RNA. So you're not going to get any information about the amount of your gene expression or anything from that. That is just simply a way to run an RNA gel to see the fluorescence peaks in before you do any amplification with gene-specific primers. What is the ratio of the 16 or the 18S and 28S ribosomal RNA, and that is a proxy for how degraded that RNA might be, but you get no information about quantification of any sort of gene, basically. Okay.
And then we have kind of a follow-up question to that. What kind of gel would you use to check RNA integrity?
Right, yeah, I see. So with these, like these RNA gels, basically, the old school way to do it is to just run a gel with your RNA and look at these two bands and say, okay, this one's a little bit brighter than this one, and I don't have any weird stuff, and I can see the two distinct peaks. The bioanalyzer RNA gel, so this is a product, right? Like this is like an Agilent product that basically they have, and it's like by far the most commonly used in the field, but basically it's a microfluidics chip. So you're not actually really, it doesn't even look like a gel. What you're doing is you're pipetting onto these little tiny microfluidic channels your sample, and then it goes into a machine and it spits this data out where you get these fluorescence peaks. So yeah, this is like, it looks fancy, but it's really easy to do, and it's a machine does all these calculations for you. So yeah, that's the RNA gel. Yeah, but you could also just run a regular gel and look at the two different peaks.
That makes sense. And then Audrey's asks if you're familiar with the qubit IQ score, and if you can comment a little bit about that for assessing the integrity of your RNA.
Yeah, I kind of talked the qubit versus the nanodrop, and I've heard different people say different things. The qubit's really good for quantifying. You have to sort of think about also the fact that in qPCR you are looking at relative ratios. So as long as you're inputting the same amount at the start of everything, these whether or not you added 50 nanograms or you added 58 nanograms of cDNA to your reactions actually doesn't have any effect on the full change values. So, however, these qubit versus nanodrop, like that's really important when you're doing RNA-seq and other NGS technologies. And so the qubit can be a little bit more quantitative. It might have a little bit more of a sensitive range for the volumes it can detect. Actually also in the bioanalyzer, you also get a calculation for the concentration of RNA that you added as well. So there's all sorts of different ways to do it. What I have used in the past is when I'm doing next-gen sequencing and RNA-seq, I will use the qubit to quantify the RNA and to quantify the libraries, as well as using the bioanalyzer to make sure that the RNA is good quality. So usually for RNA-seq, you have like, okay, we're not even going to run RNA-seq unless the RIN is above 7, and we are going to quantify how much is there using the qubit.
Okay.
Yep.
That does make sense. That makes perfect sense. And then I think we have our last question. We have 2 more questions left. So the first one is from Zane again, and they're asking about doing quantification with intercalating dyes. They are referring to number quantification.
Sorry, I think I didn't get quite the last part. So using DNA intercalating dyes, what's the question?
Okay, it says, can we do, so I'll read the exact question. Can we do the quantification with intercalating dyes, like copy number rather than relative quantification?
Well, with copy number, is that what they're asking? Like if you use plasmid standards?
I think so, yes.
Yeah, absolutely. So if you have some gold standard plasmids that you know the copy number of, and there's kits that have this, you can do it yourself. So one way to do it, for example, is to order a plasmid and linearize it, or order one of those long oligos like IDT-G blocks. And basically what you can do is then make serial dilutions of that stock, and then you can quantify the actual copy number that are in each one of those stocks. For the instead of the concentration in nanograms per microliter, you can, using the molecular weight of the size of the DNA, you can calculate the number of molecules, and then you can check that. The best way to check that actually is to use digital PCR, and then you can actually have a real, like really quantitative. So this is what I've done in the past for validating like clinical assays, is we have these copy number standards, which we have an error value on. Like we know that it's 10 copies of the target gene plus or minus 2 based on many runs in digital PCR. And so what you can then do is you run a standard curve with every qPCR run. And so then you build a linear model with that standard curve over the CT value versus the range of concentrations you've used. And then you fit your CT values to that linear model. And then that will give you, rather than using full changes, that will give you copy numbers of the gene of interest, if that makes sense. So yeah, that's a really good way to do it. It's really quantitative. So I would actually refer you, if you're interested in that, to this paper here. That's that's what I've done here. And so there's actually a lot of information in that supplement on how I went about doing that, and I put all the experimental information in there too. So
that's great. And Zane's asked a follow-up question about plasmids or standards are applicable to all kinds of samples.
Well, I mean, it depends.
Yeah.
It sort of depends. I mean, yeah, it's like whatever you want to do with the plasmid. Like if you want to show your results in terms of copy number, then use a plasmid. If you want to show your results in terms of fold change between two groups, which is what people commonly do, then yeah, you don't need a plasmid. But it definitely depends on what you're trying to do.
Okay. And then I think our last question comes from Samuel. They're asking about cDNA. You said cDNA is not quantified when you're doing RT-qPCR, but how do you determine how much cDNA should be added to each RT reaction?
Sure. So, let me step back. So cDNA is definitely quantified in qRT-PCR. That's what we're actually measuring. But what I think someone had an earlier question on was whether or not you do any QC quantification on that cDNA. So what I think people might be referring to is, okay, let's say you've made your, and that kind of goes back to this slide, like let's say you've made your, you've isolated RNA, okay, and you have your RNA concentration is whatever it is, and you make cDNA using 1 microgram of RNA, well, it's 1,000 nanograms, okay? And then you have that cDNA stock of like 1,000 nanograms per 20 microliters. Let's say that's your final reaction volume. What the practice is in the field is to just proceed with this PCR reaction as if this, as if 100% of the RNA is basically cDNA. So in fact, like that's correct. Like the amount you're actually adding is less. It's quite a bit less than 50 nanograms. Okay, if you go through this example, because not all of that cDNA is going to be actually amplified. It's a small fraction of it.
Okay. Well, that brings us to the end of the webinar. So thank you again, Matthew, for a very illuminating presentation and a fantastic discussion. And thanks also to our sponsor, Qiagen. Sorry.
Yeah, I just wanted to let people know that everything that I've shown in the analysis is it's on this GitHub. So if people want to go through and take a look at anything, let me just move this. Basically, what they can do is go to this my GitHub site, which is github.com/mattpm, and click on this qRT-PCR webinar, and everything I've shown is here with our Markdown analysis, or you can view it as a Word document or HTML, and also the PDF of the slide deck I showed if you want to refer to anything. So I just thought I'd mention that resource is there if anyone wants to use it and go through the analysis steps again, for example. Feel free to do so.
And we will also post a link to Matthew's GitHub site on the webinar page when we post the video as well, just to let everybody know in the audience.
Great.
So thanks again to our sponsor, Qiagen, and finally, thanks to you all, the audience, for taking the time to attend and listen in. If you enjoyed the webinar and would like to view a video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours or so. And there you can see the other webinars we've lined up for you on Bitesize Bio. So until next time, good luck in your research, and goodbye from all of us at Qiagen and Bitesize Bio.